Hello everyone, welcome back to Microbiosolutions. Solutions. Today we are going to discuss about competitive ELISA. So same like indirect ELISA, competitive ELISA is also used to detect the presence of antibodies in the sample. Here, unlike other ELISA, the intensity of color produced will be inversely proportional to the amount of antibody present in the sample. Remember, intensity of color is inversely proportional to the amount of antibody present in the sample. Whereas in direct ELISA, indirect ELISA or sandwich ELISA, the intensity of color produced will be directly proportional to the amount of antigen or antibody in the sample. Here, the antigen will be coated in the microtiter plate as we are detecting the presence of antibodies. If the antibodies are present in the serum, it will directly bind to the antigen. Okay. So if there are no antibodies in the serum, the antigens will be left without forming an antigen-antibody complex. Okay. Then by the addition of enzyme conjugated antibody, this enzyme conjugated antibody will go and occupy this unbound antigen which is already present in the microtiter well. So first there will be antigen in the microtiter well and the patient sample we are adding. If there are antibodies in the patient sample, it will go and bind to the antigen in the microtiter well. But if there are no antibodies in the patient serum, the antigens will be left free. Okay. So upon addition of this enzyme conjugated antibody, this enzyme conjugated antibody will go and attach to the unbound antigen. Here this won't act as an antibody towards the primary antibody. So it is going and attaching to the unbound antigen, followed by the addition of substrate here, which is specific for the enzyme conjugate for the formation of colored end products. So what happens here? If there are no antibodies in the patient serum and the antibody coated enzyme conjugate or enzyme conjugated antibody have bound to the unbound antigen, upon the addition of substrate, that will produce the color. So if there are no antibodies, the intensity of color will be more, but if there are antibodies, the intensity of color is less. That is what we have told in the first slide. The color produced or the intensity of color produced is inversely proportional to the amount of antibody present. Because if there are no antibodies, there will be more color. If there are antibodies, there will be less color. So if there are more antibodies in the serum or sample, the intensity of color produced will be very less since there are no antigens left in the microtiter plate to bind with the enzyme conjugated antibody. And if there are no antibodies, thus the intensity of color produced will be high. That's what we have told, that the intensity of color produced is inversely proportional to the amount of antibody in the sample. So seeing this picture, you will be able to identify. Consider this as the sample. This is the sample which has got antibodies and this is not having any antibodies. We'll see this sample treatment first. Okay, this is a microtiter plate with the antigen, pre-coated antigen. You are adding your sample to this microtiter well with pre-coated antigen. Okay, you have added your sample and the antibody has gained attachment or it has bound to this particular antigen. Okay, then you are washing and then adding your secondary antibody with an enzyme conjugate. This is the enzyme conjugate and then you are adding your secondary antibody. Here, as two antibodies from the patient sample has already bound to this pre-coated antigen, there is only one space or one valency in the antigen. One antigen only left for this enzyme conjugated antibody to bound. Okay, so that place this has bound and then it is taken for another wash and the substrate is added. So since the substrate is added, the substrate can only act with this antibody which has got this enzyme conjugate. So there will be less color, or light blue or it could be clear because this place or this antigens has been occupied with the antibodies in the patient sample. Okay, now we can see this sample without any antibodies. Say you have your microtiter plate with the antigen. You are adding your sample but there are no antibodies. Then you are washing. There are no antibodies. There are no antigen-antibody reaction. Then you are adding your enzyme conjugate. Since these antigens are free to get binding, Your enzyme conjugated with antibody will occupy all the spaces. Okay, you can see all these three antigens, valencies are occupied by this enzyme conjugated antibodies. So, when you are adding the substrate can react with this enzyme conjugated antibodies and the color can be produced. That is why there is a competition between the antibody present in the sample and the enzyme conjugated antibody to gain attachment to the antigen. That is why it is called as competitive ELISA. You can see the color of this is dark blue where it is light blue or clear, because the intensity of color produced is inversely proportional. You can see there are no antibodies here in the patient sample, so there is more color. There are antibodies in the patient sample, there is light color. That is the principle behind competitive ELISA. So this is a small recap between all the ELISA: direct ELISA, indirect, sandwich and competitive ELISA. Indirect ELISA, the antigen is there, you are going to detect the antigen and the primary antibody conjugate is added along with the enzyme conjugated, and then the substrate is added, there will be colored end product. In indirect ELISA, you are detecting the presence of antibody. So you have a microtiter well which is pre-coated with an antigen, then your patient sample with antibody is added, then this enzyme conjugated antibody will act as an antibody towards this primary antibody, and then when the substrate is added, there will be colored end product. Whereas in sandwich ELISA, you have a capture antibody or a monoclonally raised antibody which is pre-coated in the microtiter well, then you are adding your antigen. This is also for detection of antigen. Then you are adding your primary antibody. Okay, here this antigen is sandwiched in between these two antibodies. This it is called as a sandwich ELISA. Then next when you are adding your enzyme conjugated antibody, which is acting as an antibody to this antibody, and followed by the addition of substrate which forms a colored end product. Next is the competitive ELISA. As you have seen, there will be competition between two antibodies, that is enzyme conjugated antibody and the antibody in the patient sample, to bind to the antigen which is pre-coated in the microtiter well. And upon the addition of substrate, if there are antibodies in the patient sample, there will be less intensity of color, and if there are no antibodies in the patient serum, there will be more intensity of the color. This is about ELISA and this is about today's lecture. If you have any queries, please post it in the comment section. Please do like, share and subscribe to our channel. Thank you.